Cyclin is a family of proteins that control the progression of cells through the cell cycle by activating cyclin-dependent kinase (CDK) enzymes. Etymology: cyclins were originally discovered by R. Timothy Hunt in 1982 while studying the cell cycle of sea urchins. In an interview for The Life Scientific, aired on 13 December 2011, hosted by Jim Al-Khalili, R. Timothy Hunt explained that the name cyclin was originally named after his hobby cycling. It was only after the naming did its importance in the cell cycle become apparent. As it was appropriate, the name stuck. R. Timothy Hunt. By the way, the name cyclin, which I coined, was really a joke. It's because I liked cycling so much at the time. But they did come and go in the cell. Quote, function. Cyclins were originally named because their concentration varies in a cyclical fashion during the cell cycle. Note that the cyclins are now classified according to their conserved cyclin box structure. And not all these cyclins alter in level through the cell cycle. The oscillations of the cyclins, namely fluctuations in cyclin gene expression and destruction by the ubiquitin-mediated proteasome pathway, induce oscillations in CDK activity to drive the cell cycle. A cyclin forms a complex with CDK, which begins to activate the CDK, but the complete activation requires phosphorylation as well. Complex formation results in activation of the CDK active site. Cyclins themselves have no enzymatic activity but have binding sites for some substrates and target the CDKs to specific subcellular locations. Cyclins, when bound with the dependent kinases, such as the PE34, CDC2, CDK1 protein, form the maturation-promoting factor. MPFs activate other proteins through phosphorylation. These phosphorylated proteins, in turn, are responsible for specific events during cycle division such as microtubule formation and chromatin remodeling. Cyclins can be divided into four classes based on their behavior in the cell cycle of vertebrate, somatic cells and yeast cells. G1 cyclins, G1, S cyclins, S cyclins, and M cyclins. This division is useful when talking about most cell cycles, but it is not universal as some cyclins have different functions or timing in different cell types. G1, S cyclins rise in late G1 and fall in early S phase. The CDKG1, S cyclin complex begins to induce the initial processes of DNA replication, primarily by arresting systems that prevent S-phase CDK activity in G1. The cyclins also promote other activities to progress the cell cycle, such as centrosome duplication in vertebrates or spindle pole body in yeast. The rise in presence of G1, S cyclins is paralleled by a rise in S cyclins. G1 cyclins do not behave like the other cyclins, in that the concentrations increase gradually, with no oscillation, throughout the cell cycle based on cell growth and the external growth regulatory signals. The presence of G cyclins coordinate cell growth with the entry to a new cell cycle. S cyclins bind to CDK and the complex directly induces DNA replication. The levels of S cyclins remain high, not only throughout S phase, but through G2 and early mitosis as well to promote early events in mitosis. M cyclin concentrations rise as the cell begins to enter mitosis and the concentrations peak at metaphase. Cell changes in the cell cycle like the assembly of mitotic spindles and alignment of sister chromatids along the spindles are induced by M cyclin CDK complexes. The destruction of M cyclins during metaphase and anaphase. After the spindle assembly checkpoint is satisfied, causes the exit of mitosis and cytokinesis. Expression of cyclins in individual cells in relation to cellular DNA content. Cell cycle phase is often measured by flow cytometry. Their expression is also being measured by flow cytometry concurrently with the initiation and termination of DNA replication during S phase. Domain structure. Cyclins are generally very different from each other in primary structure or amino acid sequence. However, all members of the cyclin family are similar in 100 amino acids that make up the cyclin box. Cyclins contain two domains of a similar all-alpha fold, the first located at the N-terminus and the second at the C-terminus. All cyclins are believed to contain a similar tertiary structure of two compact domains of 5-alpha helices, the first of which is the conserved cyclin box, outside of which cyclins are divergent. For example, the amino terminal regions of S and M cyclins contain short destruction box motifs that target these proteins for proteolysis in mitosis. Types. There are several different cyclins that are active in different parts of the cell cycle and that cause the CDK to phosphorylate different substrates. There are also several orphan cyclins for which no CDK partner has been identified. For example, cyclin F is an orphan cyclin that is essential for G2/M transition. A study in C. elegans revealed the specific roles of mitotic cyclins. Notably, recent studies have shown that cyclin A creates a cellular environment that promotes microtubule detachment from kinetochores in pro-metaphase to ensure efficient error correction and faithful chromosome segregation. Cells must separate their chromosomes precisely, an event that relies on the bi-oriented attachment of chromosomes to spindle microtubules through specialized structures called kinetochores. In the early phases of division, there are numerous errors in how kinetochores bind to spindle microtubules. The unstable attachments promote the correction of errors by causing a constant detachment, realignment and reattachment of microtubules from kinetochores in the cells as they try to find the correct attachment. Protein cyclin A governs this process by keeping the process going until the errors are eliminated. In normal cells, persistent cyclin A expression prevents the stabilization of microtubules bound to kinetochores, even in cells with aligned chromosomes. As levels of cyclin A decline, microtubule attachments become stable, allowing the chromosomes to be divided correctly as cell division proceeds. In contrast, in cyclin A deficient cells, microtubule attachments are prematurely stabilized. Consequently, these cells may fail to correct errors, leading to higher rates of chromosome missegregation. Main groups. There are two main groups of cyclins. G1S cyclins, essential for the control of the cell cycle at the G1S transition. Cyclin A, CDK2 active in S phase. Cyclin D, CDK4. Cyclin D, CDK6. And cyclin E, CDK2 regulates transition from G1 to S phase. G2/M cyclins, essential for the control of the cell cycle at the G2/M transition. Mitosis. G2/M cyclins accumulate steadily during G2 and are abruptly destroyed as cells exit from mitosis. At the end of the M phase, cyclin B, CDK1 regulates progression from G2 to M phase. Subtypes. Specific cyclin subtypes include other proteins containing this domain. In addition, the following human protein contains a cyclin domain: CNTD1. History. Leland H. Hartwell, R. Timothy Hunt, and Paul M. Nurse won the 2001 Nobel Prize in Physiology or Medicine for their discovery of cyclin and Cyclin-dependent kinase. Eukaryotic linear motif. Resource motif class lig_cyclin_1. Further reading. Monty Krieger, Matthew P. Scott, Matsudera, Paul T. Lodish, Harvey F. Darnell, James E. Lawrence Zipersky, Kaiser, Chris. Arnold Burke, 2004. Molecular Cell Biology. 5th ed. New York. W. H. Freeman and C.O. ISBN 0-7167-43663. This article incorporates text from the public domain PFAM and Interpro IPRO 06671.